You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is October 30th, 2023. Our topic is How the Immune System Works by Dr. Lauren Sompayrac, chapters 5 through 7, presented by Dr. Nikita Raje. She is an associate professor of internal medicine pediatrics and is the chief in the section of allergy and immunology at Children's Mercy Hospital.
Everyone, I'm Nikita Raje from Children's Mercy, University of Missouri, Kansas City. Today is October 30th, 2023, and we'll be talking about chapters 5, 6, 7 from How Immune System Works, textbook by Lauren Sompayrac. Feel free to stop me anytime if you have questions. Let's see if this works. Let's see. All right, so the learning objectives today are to improve the understanding about T cell activation, T cells at work, and then secondary lymphoid organs. So T cell activation, let's look at several aspects of these two different types of T cells, T helper cells and cytotoxic T cells. And we'll start with receptor and antigen binding. Okay, so antigen binding to these T cell receptors involve the T cell receptors that are two different types. The alpha beta type, which are 95% of the T cells that have alpha beta TCRs, versus 5% of the cells have gamma delta T cells. These are typically made by gene recombination, just like we talked about with B cells. And we know that there are some proteins that initiate the splicing of the gene segments. Does anyone know what proteins take part and start that splicing of gene segments so that the recombination can occur in the DNA of the developing T cells so that the TCRs are formed? Any idea?
I think they're the RAG ones, RAG1 and RAG2.
That's right, RAG1 and RAG2. So these are really important proteins that will help in this gene recombination process. So the alpha beta TCR are present on these traditional T cells, and these are diverse, extensive diversity, so that these T cells can react to various types of antigens in the environment, in the nature. These T cells are trained in the thymus, and they recognize the peptide that is bound to MHC molecule on their surface. They also have some co-receptors that help in activation. So, for example, CD4 and CD8 are the two different types of co-receptors that can be present on these alpha beta T cells. But unlike the B cells that undergo hypermutation, these T cells do not undergo any hypermutation. So there is no somatic hypermutation after they are activated. The non-traditional T cells are gamma delta. These are just make up the 5% of the T cells, and these TCRs are less diverse. They are found in the thymus, but they can be functional even without the thymus, and they do not need MHC for presentation to them. So there are different types of cells that can present antigens to them, and these cells will recognize stress proteins like MICA, MICB. There are no co-receptors on these cells, so there's no CD4. There's no CD8 on their surface, so these would be included in the double negative T cells, which make up about 5% of the T cells. These are present anywhere that the body comes in contact with outside. So intestine, uterus, tongue, all these places will have gamma delta T cells. 1% of the T cells are NK T cells or natural killer T cells, which means they have markers for NK cells as well as the markers for T cells. They also have alpha beta TCR on their surface, but somehow these alpha beta TCRs have limited diversity, unlike the other alpha beta T cells. And they recognize lipids by a non-classical MHC. Anyone know what the name of that MHC is?
CD1.
So CD1 is it has a structure similar to MHC, but instead of presenting peptides, it can present lipids to these T cell receptors. All right, so for T cell activation, there needs to be several different steps. We looked at the binding portion, which is the receptor and antigen binding, right? Now we talk about the signaling molecule. Why is this important? Because the TCR itself, say alpha beta, that's our traditional T cells. Here's a cell, that's a T cell. Here's a cell membrane. The alpha beta TCR is attached to the cell membrane. But if you see intracellularly, it doesn't have a long chain. So what that means is if the signal has to go to the inside of the cell, we need some other molecules that can help these T cell receptors. So the whole point of the signaling or T cell activation is once it's activated, this antigen and this receptor that bound to the antigen can transmit the signal of activation to the inside of the cell or the nucleus so that there is activity that's created based on that activation. But to do that, that signal needs to reach the inside, and the TCR is pretty short to do that. So there are signaling molecules in vicinity of the TCR. So typically that involves various different chains that are present in close proximity to the TCR. So the intracellular protein that has some of these chains is called CD3, and that includes all these chains called gamma, delta, epsilon. So gamma, delta, and epsilon chains together form the CD3 along with the zeta chains. All of these chains together provide that signaling that's required that can take that activation signal from the surface to the inside of the cell. So the TCRs can also cluster like the BCR that we had talked about last time. Remember, all these BCRs on the surface or the TCRs on their surface, if they come close to each other and attach to the same antigen, then they kind of get clustered so that will help in activation. Also, there are some enzymes that get recruited to the cytoplasmic tails of these chains so that there is signaling. Now, the signaling molecule can be a single or a two chains, just like the BCR had that Ig alpha and Ig beta attached to it. But T cells are way more complex. They have all these chains, and there is some meaning to having all these chains for signaling. And so based on what chains or how many chains, how many of those kinase enzymes are recruited to the TCR can have various different forms of activation. So the activation signal goes from the TCR, but depending on what goes on in these signaling chains, the T cell can undergo activation, or it may not show any reaction, which is called energy, or it may undergo death. So that the death typically occurs in the thymus. So say in the thymus, when the T cell is being developed and it comes across a self-antigen that's presented to it by thymic cells. The self-antigen is presented as a test to these T cells to see if it can react to a self-antigen. If it does actually react and like gets the TCR can bind to that self-antigen and it can send a signal through the TCR, in that case, that's a dangerous T cell which can cause autoimmunity because it's reacting to self-antigen. In those cases, the way the signaling works is that T cell will undergo death. Versus if this T cell is well developed, the TCR is in place and it's no longer in the thymus, it's a normal T cell that goes into the periphery, comes across an antigen in the periphery. In those cases, that antigen will activate the T cell and the signaling molecules. That signal from the TCR will go through the signaling molecules so that it can cause activation or energy. And we'll talk about these concepts more in detail. But just so that you know, there's a lot of versatility of these TCRs thanks to these signaling molecules. And so signaling chains include, remember, gamma, delta, epsilon, and zeta chains. Now, this can be confusing because this is an alpha beta TCR belonging to a traditional T cell. But there are those non-traditional T cells that will have gamma delta TCR. And the gamma delta TCR has nothing to do with the gamma delta in the signaling molecules. So just so that you know that you have to be clear that we are talking about alpha beta TCR containing T cell. The signaling chains in those are CD3 zeta chains, and that includes gamma delta chains. Those are not the surface TCR. All right, what else do we need for activation? So T cell is very picky, has lots of needs on when it gets activated and sends those signals through. So we talked about the first signal, which is the receptor binding to the antigen. That signal goes through the signaling molecule. But next to the receptor is the co-receptor. And the co-receptor actually binds to the same antigen as the TCR does, but the epitope, the part of the antigen it attaches to, is different from what attaches to the receptor. So let's see what these are. Co-receptors are either CD8 or CD4. So let's look at these helper or cytotoxic T cell. Helper T cell binds to MHC class 2 versus cytotoxic T cell can bind to MHC class 1. So cytotoxic T cell has the co-receptor CD8 on it versus the helper T cell has CD4 on it, right? So CD4 can attach to MHC class 2, CD8 can bind to MHC class 1. So remember again, the TCR is the one that binds to the peptide and the MHC class 1 from the APC, right? That's your first signal. Same thing with the helper T cell binding to the TCR on the CD4 cell, binding to the peptide and MHC class 2 that's presented to it by the APC. However, you see the CD4 or CD8 co-receptors that clip onto the MHC molecule. So they interact with the antigen itself and the MHC molecule, CD4 binding to MHC class 2 and CD8 binding to MHC class 1. And they're loosely bound to the TCR along with the CD3. Remember, CD3 is our signaling molecule. So all the whole complex is kind of attached to each other and the peptide itself. The whole overall complex is important, so because it stabilizes this whole receptor antigen complex that includes the T cell receptor itself, peptide antigen, along with the MHC class 2. And the co-receptor also helps in strengthening the TCR signal that goes through the CD3. So based on the based on the CD4 or CD8 co-receptors in the thymus, the developing T cell goes through various stages of maturation. It starts out as a thymocyte. So remember the lymphocytic progenitor that are produced in the bone marrow go to the thymocyte, and the ones that go to the thymus, and the ones that go to the thymus are called thymocytes. They do not have CD3 on them, they do not have CD4 on them, they do not have CD8 on them, but they are destined to become a T cell. So those cells are called thymocytes, and over the period of maturation, they go through various stages as they get these markers. The first one they'll get is a CD3, and then as they develop, they do not have either CD3 cells do not initially have CD4 or CD8 on them. So they are called double negative T cells. The double negative T cells then will interact with MHC, and they will actually have both CD4 and CD8 on them, which are called double positive T cells. And then eventually, based on what they come across, the MHC class 1 or class 2, with their interaction, they decide whether they want to be a CD4 or a CD8 cell. So you'll see that some cells, the T cells, will become CD4 positive T cells, single positive cells, or CD8 positive, single positive T cells. Both of these are CD3 positive as well. So the choice of the co-receptor is determined by the class of MHC that the TCR prefers or just interacts with. So remember again, this is the maturation of T cells in the thymus based on which the co-receptors are develop on the surface of the T cells. So even that's not enough for activation, and the T cells need one more step before they'll get activated. And that's called the second signal or the co-stimulator use. So let's talk about co-stimulation. So what are naive T cells? So naive T cells are mature T cells. These are single positive T cells that have gone through all those maturation steps that we just talked about. So thymocytes arrived in the thymus, went through the double negative and then double positive, and then came across MHC and decided to be either a CD4 or a CD8 positive T cell. All of those mature cells, T cells, are called naive T cells, which means that they have not come across the antigen that they react to or bind to. So once the T cells interact with the antigen that they can bind to, they get activated, and at that time they are no longer called naive T cells. So naive means that they are new and are inexperienced, and that's what the meaning of naive T cells is. They are inexperienced, they have not come across the antigen that they can bind to. So what's the role of co-stimulation in these naive T cells? So naive T cells, when they come across the antigen they can bind to, they're going to interact with it and bind to it and hopefully get activated. But the co-stimulation reduces the number of TCRs needed to be cross-linked to activate naive T cells about 100-fold. So say there's a B cell that's binding to pneumococcal antigen. In that case, the BCRs get cross-linked because they bind to multiple of the epitopes present in a polysaccharide. In one layer, there are several epitopes. And so the several BCRs bind to those epitopes, so they are cross-linked, and there's clustering. But when it comes to the T cell, that might not occur as quickly. And so it's easier if there is another way of helping activate these T cells and giving a second signal. That second signal is provided by the co-stimulation. It strengthens the connection between TCR and the nucleus once it's activated. So what provides this co-stimulation? The T cells have a marker called CD28 on their surface. When the APCs present the antigen peptide on the surface of the MHC class 1 or class 2, that will interact with the TCR on the T cells. The co-receptor CD4, CD8 will also bind to the same antigen and the MHC. However, the second signal is provided by APCs having B7-1 or B7-2 molecules on their surface that interact with CD28 on the surface of T cells. And so that provides a signal to the T cells that there is danger. And that's when the T cells get activated. So that's your co-stimulatory signal for the T cells. Now, interestingly, say a T cell was activated and now it goes into the periphery and it's a memory T cell. It actually needs less of a co-stimulation than a naive T cell to get activated again. All right, so let's talk about helper T cell activation. Um, So we are going to go through the same kind of interaction that we talked about. A naive T helper cell comes across an APC, like a dendritic cell. Adhesion molecules keep them together on both these cells, right? So that's your peripheral area where the adhesion molecules are just keeping them together. And then the class 2 MHC interacts with CD4. And the TCR interacts with the cognate antigen or the peptide bound to the MHC class 2. And then this results in formation of uh, a, an area on these two surfaces that's called immunological synapse. So that area where all these molecules on the surface of both the cells come in contact with each other. That whole area is called immunological synapse, where most of the interaction and most of the activity takes place. All right, so in general, that's the process, but here is one more information on another co-stimulatory molecule. So say a TCR binds to the cognate antigen. It also upregulates a molecule on its surface called CD40 ligand. And CD40 ligand will interact with CD40 either on the APC. That APC can be a dendritic cell or a macrophage or an activated B cell. After that interaction, the dendritic cell increases MHC and B7 expression on the dendritic cells and prolongs the life. That interaction also prolongs the lifespan of the dendritic cell or any other APC that has CD40 that interacts with CD40 ligand on its surface. Similarly, for activated B cells, they will have CD40 on their surface, and the high levels of CD40 ligand on T helper cell activates B cells. So this interaction also is a co-stimulatory molecule interaction similar to CD28, B7-1, B7-2. Once the activation occurs, the dendritic cell gets disengaged. That synapse breaks so that it's disengaged from the activated helper T cell. Activated helper T cells produce IL-2, and the activated helper T cells also have IL-2 receptors on their surface. So what happens is, because it produces IL-2 that acts on the same cell, it will stimulate its own proliferation, and so it gives rise to multiple T cells with the same kind of TCR, so that they form a group of cells called a clone, and that's called clonal selection. Similar to helper T cells, there is similar interaction on the cytotoxic T cells. Activated dendritic cells will interact with naive cytotoxic T cell. That happens in the early part of activation. In the later part, actually, the helper T cells also help in the activation of cytotoxic T cells. So there is a three-way interaction between these three cells: helper T cells, antigen presenting cell, and the naive cytotoxic cell. Same kind of interaction. TCR binds to the cognate antigen, and the MHC class 1 binds to CD8, so the co-receptor, and then co-stimulation. So the receptor, co-receptor, and then co-stimulation. All three steps need to occur for that activation. And then activated helper T cell can release IL-2. That IL-2 will help in proliferation of cytotoxic T cells. Because of this, there is efficient killing, and the cytotoxic T cells become long-lived, and they can turn into memory T cells. So that's the part of activation that we wanted to talk about. That was the first chapter. Let's talk about what happens after activations. So after activation, T cells are at work, and so they are called effector T cells. So the activated T cells have to differentiate into effector T cells so that they can work. So effector T cells are working under the guidance of their coach, which are the antigen presenting cells or dendritic cells or macrophages, right? So let's talk about what kind of effector T cells we are talking about. These could be helper T cells or cytotoxic T cells. Helper T cells can be of different subsets based on what kind of work is at hand. They can become helper T cells subset 1, 2, 17, or Tregs. There are also some helper T cell subsets that do not differentiate initially, and they are called Th0. So here are Th1, Th2, Th17, Th0 subsets, and then Tregs are another subset as well. All right, so how do the coach cells, which means the dendritic cells, decide and help the activated T cells to become different types of subsets of helper T cells? So remember, the dendritic cells are a cell that will take the information about what type of infection is it. Is it a viral infection? Is it a bacterial infection, fungal infection? They take that information from that site of infection and take it to the lymph node where they interact with these activated cells. And they themselves are the ones that are going to present that antigen to activate the T helper cells. So how do they get this information? So the dendritic cells that are present at the site of infection, on their surface, they have pattern recognition receptors like toll-like receptors, and they all have cytokine receptors. So they are in presence of whatever activating signal it is. It depends on a viral infection or bacterial infection. They take that, they use their pattern recognition receptors to get activated to know what type of bug is present in that area. And they also recognize the cytokines that are released at that site, say by macrophages, and the cytokine receptors interact with the cytokines present there, and they take that information. And they take that information and go to the lymph node and activate the T cells and provide the message of the type of infection that's going on. That helps the T cells to get the information of what type of helper T cells are needed, and they will differentiate into that type of helper T cell subset. Another type of information that these dendritic cells provide is the site of infection or the invader. And that information is also taken up by the cytokines that are released. So the cytokines that are released in the gut, if there is an infection, versus the cytokines that are released in the skin may be different. And that information, because the cytokines receptors that are used up and what cytokines they can release are different, those dendritic cells go to the lymph nodes, activate the helper T cells, and in that process, provide the information of the type of infection and the site of infection to these helper T cells. And that helps them to decide what kind of effector T cells are going to be produced. So so the T cells are definitely the quarterback who are doing all the work, but they get all that information from their coach, dendritic cells. These two, pattern recognition receptors and the cytokine receptors, the cytokines that they release, all of that is used by dendritic cells to provide the information, to collect the information that they're going to provide. But that information is transferred to the activating T cells using the co-stimulatory molecules that they use and the cytokines that they release. That information is using the co-stimulatory molecules and cytokines is passed on to the helper T cells, and that's how they help the T cells in their job. So let's take a look at how that happens in each of these subsets. So think about a viral infection. Virus is a intracellular organism. So keep that in mind. If there is an intracellular organism such as virus or, for example, mycobacteria, in those kinds of infections, the dendritic cell comes across that virus or bacteria, uses its toll-like receptors or other pattern recognition receptors, and recognizes that it's a virus or it's a mycobacteria. And it takes a look at the cytokines that are present at that place. So say in case of a virus, there might be interferons in that area. Takes that information and it activates the helper T cells and releases cytokine IL-12. In the presence of IL-12, this interaction between TCR and MHC class 2 and peptide will end up in activation of the helper T cell that will release cytokines interferon gamma, TNF, and IL-2. All these cytokines are going to work in a way that will help in fighting the infection that's caused by intracellular organisms like virus or mycobacteria. So this cell that got activated was primed to become a Th1 type of cell that releases these particular cytokines. So the TNF is important in activating macrophages and NK cells. Guess what the macrophages and NK cells do? They help in fighting intracellular organisms. These macrophages will go to the site of infection and phagocytose the microbes. Natural killer cells will help in fighting against the cells that are infected with virus or those intracellular microbes. Interferon gamma keeps the macrophages activated and will help in class switching B cells to IgG3 that helps in fighting these infections. And then IL-2 helps in activating NK cells and improve killing of those by those cells, and they help in proliferation of the cells such as NK cells, cytotoxic cells, and the helper Th1 type of cells. So the end result is that the cells that are needed to kill this particular microbe are activated and are functional because of the cytokines released by a helper T cell. Now, compared to that, let's think of another type of infection. Say it's a parasitic infection, or there is ingestion of a pathogen in the gut. At that place, when there is a parasitic infection, there are cytokines released in that environment like IL-4. In presence of that IL-4 cytokine, the dendritic cells will use their pattern recognition receptors, come across that microbe and take that information of the type of infection, which is parasitic infection, and the site of infection, say the gut. It will take that information along with that and prime to activate these helper T cells that they come in contact with and help those helper T cells to release cytokines such as IL-4, IL-5, and IL-13. And these cytokines are needed to fight the particular infection at hand, which is like a parasitic infection. So the IL-4 that's released is a growth factor for Th2 cells, so it helps in positive feedback. It's also a growth factor for B cells and helping class switching to IgE here. Remember, Th1 used it to produce IgG. Here, it's IgE that's produced. IL-5 that's released helps in class switching to IgA. That's important for gut, as well as IL-13 that releases mucus in the intestines. So all these cytokines that were released because of the information that dendritic cells provided the helper T cells will result into formation of or activation of cells that are important for fighting parasitic infection. So that's how the dendritic cells provide the information to the helper T cell so that they become Th2 type of effector T cell. Third example is an extracellular bacteria. Remember, Th1 was for intracellular microorganisms, whereas the Th17 are for extracellular bacteria or fungus. In that kind of infection, the dendritic cells use their pattern recognition receptors to realize that it's, say, a fungal infection or an extracellular bacteria like strep, right? In those cases, it's going to release cytokines TGF-beta and IL-6 that act on the helper T cell to make Th17 cells. Th17 cells give rise to IL-17 and IL-21. IL-21 helps in leading Th0 cells to form more Th17 cells and help in class switching to IgG. Th17 helps in recruiting neutrophils to the site of infection and class switching to IgG. And the IgG as well as neutrophils help in fighting what kind of infections? Fungal infections and extracellular bacteria. So the dendritic cells led to the formation of effector molecules that are important for fighting the infection at hand and gave that information to the helper T cell so that that particular type of helper T cell will produce cytokines so that these molecules will be formed, IgG and neutrophils. All right, what do Th0 cells do? So dendritic cells, similar to the other things that we looked at, Th1, Th2, Th17, will activate the Th0 cells. They'll give the information to them about the site of infection, but they will not provide what type of infection, what is the microbe. So what these Th0 cells do is they actually go to the site of infection because now they are Th0 cells. They know where to go because they know what that infection, where that infection is. But when they reach that site of infection, they realize what cytokines are present around them. And based on that Th0 cell can be malleable. It can become a Th1 cell, it can become a Th2 cell. So there are some infections where you may see two different types of cytokines released, and the Th0 cell can become one or the other type of cell. All right, so these Th0 cells sometimes reach the site without commitment. And when they go to the site, the macrophages in the area may be releasing IL-12 because there is a viral infection or a mycobacterial infection, for example, and they'll tell this IL-12 will be used by these macrophages to tell the Th0 cells to form a particular type of helper cell, such as Th1 cell. If it was IL-4 and IL-5, this Th0 could have become a Th2 cell. Or if there was a fungal infection, the Th0s at that site could become a Th17 cell. Once that Th cell profile is created, it provides feedback to its own self, so it provides growth factor for its own proliferation and growth. Interestingly, it also provides negative feedback so the other subsets are not formed. So it will help in proliferation of its own self, but it will prevent proliferation of other type of helper subsets. So interferon gamma that's released by Th1 subset will actually prevent formation of Th2 cells, and IL-10 might actually prevent formation of Th1 cells. But as you can see, there is some malleability. So there is flexibility of the simultaneous defense against different invaders. So the cytokines that are released have a local action at the site of infection. So say there is a skin infection going on by a bacteria, say a staph infection that's going on in the skin. The Th cells that go there get activated, become, say, Th17 cells and start helping with fighting that infection. At the same time, someone could have a GI infection, and at that place, you don't want those Th17 cells to be present. You might want Th2 type of cells because there is a parasitic infection. So there can be two different infections going on, and there can be two different responses from the helper T cells that could be going on at different places. All right, so that was T helper cells. Now we talk about effector T cells. So, effector cytotoxic T cells. So effector cytotoxic T cells also have similarities in how they get activated and form the immunological synapse. So that process stays the same, which means there are adhesion molecules that keep the antigen presenting cell and cytotoxic cell, T cell activated to be in proximity to each other. Then there is the TCR and MHC class 1 with the antigen binding to the TCR on the CTL. Also, there is a co-receptor, so CD8 binding to class 1 MHC and co-stimulation. All of that occurs so that the T cell is activated, and then it differentiates into an effector cytotoxic T cell. The effector cytotoxic T cell, because it got activated, now has different enzymes present in it, the machinery it needs to do its job. So once it's activated, the cytotoxic T cell is ready with all the machinery it needs to perform its function. So the enzymes that are needed are called perforin and granzyme. The perforin and granzyme, the way they work is in that immunological synapse, the cell that presented the antigen to the cytotoxic T cell, the cytotoxic T cell was able to confirm that it's a danger. So the perforin is released in that immunological synapse, and that makes a hole on the target cell, on that antigen presenting cell. In case of CTLs, the antigen presenting cell is the infected cell. So that cell now has a hole created by perforin, and the granzyme is released into the cell. The granzyme B that's released leads to an enzymatic chain reaction that leads to apoptosis of that target cell. So that's one mechanism by which cytotoxic T cells kill their target cells. The other mechanism is provided by Fas ligand. Fas ligand is present on cytotoxic T cells so that it can bind to Fas that's present on the target cell and lead to apoptosis as well. And then the vesicles that are cleared by the macrophages. So this is just showing how all these cells interact and how each of these cells will use different kinds of co-stimulators, and once they get, that helps in their activation. And then they also create different types of helper T cells that lead to the formation of subsets that are needed to kill that particular microbe. All right, moving on to the next chapter, secondary lymphoid organs and lymphocyte trafficking. You would think that, the important parts are T cells and B cells and how they fight infections, but really there is something going on in background that we really need to be aware of that provides the medium in which all of this information is being used and all that activity is occurring. And that's provided by the secondary lymphoid organs and how the lymphocytes move from one place to another. So there are two types of lymphoid organs, primary and secondary. Primary lymphoid organs, thymus and bone marrow, as well as secondary lymphoid organs, which are lymph nodes, spleen, mucosal associated lymphoid tissue, all of those. So what's the purpose of having all this lymphoid tissue? So in general, there are three phases of defense. First thing is recognition of danger. The second is, now that it's recognized that there is danger, there should be production of appropriate weapons to fight that infection. And even if it's produced, that weapon needs to reach the site of infection for those weapons to work. So the transportation of the weapons to the site of attack is really important as well. So with that in mind, let's talk about these different organs. We are not going to talk about the secondary lymphoid organs. So to understand their function, the first step is to understand their structure. So the structure of lymphoid organ, that's a typical structure in some of these secondary lymphoid organs, is called lymphoid follicle. They also have another structure called high endothelial venule, but there is an exception to that, and we'll come to that. Keep thinking of what secondary lymphoid organ does not have high endothelial venule. All right, so the lymphoid follicles are primary or secondary. And the lymphoid follicles are typically areas of the secondary lymphoid organ where there are B cells. So these are B cell structures. So the initial lymphoid follicle, that's a primary follicle, has follicular dendritic cells and B cells. Now remember, the dendritic cells that are present that we just talked about with T cells are different from the follicular dendritic cells that have nothing to do with presentation to T cells. These are follicular dendritic cells that actually interact with B cells. Follicular dendritic cells are produced in second trimester and they are present in secondary lymphoid organs. They are star-shaped cells and they display antigens to the B cells. They have nothing to do with the dendritic cells that we talked about with T cells. So these star-shaped cells in the follicle actually have receptors for complement, which means they will bind to complement opsonized antigen. So from this, say there is blood circulation, there are antigens that are opsonized by the complement. That complement attached to the antigen comes into the secondary lymphoid organ and brings that complement attached to the antigen to the follicle. And in the follicle, it's plucked. The FC receptors bind to the complement receptor along with, will bind to the complement along with the antigen. So it will pluck it off that. And that way it has the antigen attached to its surface. Another way is it has FC receptors, and the FC receptors can bind to antibody opsonized antigen. Regardless of which way it will, what receptor it uses, now it has antigen attached to its surface, whether it's using complement receptors or FC receptors. Because it has that antigen attached to it, it will display that to cross-link the B cell receptors. So similar to dendritic cells presenting to T cells, these FDCs will present that antigen to the B cells so that the B cell will use its BCRs and cross-link the BCRs to get activated. Once the B cell is activated, the follicle grows and develops a germinal center, and then it's called a secondary follicle. So in the secondary follicle, there are B cells proliferating extremely fast. They double every 6 hours, and those B cells are fragile. So as they are proliferating, they need a signal to rescue them from apoptosis. If they do not get that signal, those B cells will die very quickly. So the signal they need is given to them by follicular helper T cells or T helper cells in the form of, say, CD40 ligand interaction. That will rescue them from being killed. So the B cells that survive continue to proliferate at an extremely high rate. And then those B cells that got activated will form plasma cells and leave the germinal center. So there is a center that is formed in the secondary lymphoid organ where the B cells are proliferating and are being tested against the antigen. The testing occurs in the light zone and the proliferation, extreme proliferation, occurs in the dark zone. As they move towards the periphery, they are tested against the antigen, and if they are able to survive, they'll survive or they'll continue to die. So there are two types of processes occurring in this area called somatic hypermutation, which means the FAB region of the BCR is undergoing mutation to see if they can bind in a better fashion to the antigen it's coming across. If it does, then it can improve the affinity with which it can interact with the antigen. And it can also class switch from making IgM to IgGA or E. All of this is occurring in secondary lymphoid organs, secondary follicles in the secondary lymphoid organs. The other structure in these lymphoid organs is high endothelial venule. All the secondary lymphoid organs have HEVs except for spleen. What's the purpose of HEVs? Basically, HEVs is the area of the venule where there are high endothelial cells compared to the rest of the endothelium. And in that those are the entry points that allow lymphocytes to wriggle out of the blood vessel into the lymph node or lymphoid tissue, right? Secondary lymphoid organ. And there are about 10,000 lymphocytes wriggling out into the secondary lymphoid organ from the blood circulation every second. So there are lots of lymphocytes that are getting into the secondary lymphoid organs using the high endothelial venules. So let's look at lymph node. Lymph node has two types of plumbing. It has the blood circulation from arterial and going out through venule. And it also has the lymphoid circulation, so incoming lymphatics and outgoing lymphatics. So how does the lymph entry of the cells, so lymphocytes, and entry of the antigen occur? So we just heard about how the incoming lymph brings through the HEVs will have, so sorry, let's talk about it again. So the arteriole is bringing the blood into the lymph node, and then it turns into a venule, and then there are high endothelial venules that are present in that venule where the lymphocytes are entering, lymph and lymphocytes are entering the lymph node from the circulation. However, the lymphocytes are also entering through the lymph, coming into the lymph node. The lymphocytes exit only through the lymph. The lymphocytes don't exit through the venule anymore. They are just going out through the outgoing lymph. Where are the antigens coming from? The antigens come from the lymph. So the lymphatic vessels are the ones that are bringing in the antigens that might be attached to complement or antibody when they are opsonized, or the dendritic cells that are that have processed those antigens, they bring in the antigens as well. Once it comes into the lymph node, this antigen flows, or the lymph flows from the outer cortex to the paracortex. Cortex is the region where there are, or the paracortex is the region where there are T cells, and cortex is the region where there are lymphoid follicles, and the lymphoid follicles are B cell regions. So the B cell region is the follicles in the cortex, and T cell area is the paracortex next to it. So again, arterial, high endothelial venule, and then venule. High endothelial venule releases lots of lymphocytes into the lymph nodes, and the incoming lymph brings in lymphocytes as well as the antigen bound to dendritic cells or complement and antibodies. All right, there are also some macrophages that are present in the marginal sinus that takes up big antigens when they are brought in through the lymph. So what happens? How do these lymphocytes know where to go in the lymph node? So FDCs, follicular dendritic cells, release a cytokine called CXCL13 that attract cells that have CXCR4 on their surface, and that attracts naive B cells to that area that are the follicles. So once they are come across the antigen bound to, say, complement or antibody on that follicular dendritic cell, the B cell gets activated, and then the activated B cell will downregulate CXCL13 receptors and upregulate CCR7 on its surface. Once the CCR7 goes up, it starts to move towards the paracortex, the T cell area. Activated helper T cells, on the other hand, will the naive T cells, on the other hand, that are entering the lymph node will express CCR7, so they directly go to the paracortex area or the T cell area. And when they get activated, they downregulate the CCR7 and start upregulating CXCR5. So the CXCR5 takes them closer to the follicle attracted to CXCL13. So the B cells initially express CXCR5 and go to the follicular area because they are attracted to CXCL13. Then they downregulate. Once they are activated, they have to move towards the T cell area so that they downregulate CXCL13 and upregulate CCR7, the T cells do the opposite. So the end result is that the T and B cells start to crawl towards the chemokines that they have receptors for and move towards each other so that the T cells and B cells can interact with each other and talk to each other, right? So that's what the T cells do. The T cells will express CD40 ligand, B cells will express CD40. They bind to each other and activate each other. So the follicular dendritic cells pretty much pluck the antigens from the follicular dendritic cells so that they present that to the B cells. They pluck the antigens, and that plucked antigen is expressed on the B cell surface using MHC class II. Here, the B cell is acting as an antigen presenting cell to this helper T cell. TCR binds to that and then expresses CD40 ligand so that it can help and activate B cells. With that interaction, along with another co-stimulator such as ICOS, so ICOS binds to ICOS ligand, and that also helps further in T cells helping B cells in their function. So these helper T cells in the follicle are called follicular helper T cells. Remember, these are the cells that help in rescuing B cells from apoptosis. Now that they are activated, and they help in somatic hypermutation and class switching so that the B cells become really active and basically become really prominent and functional to produce antibodies with high affinity and class switched to different kinds of classes that are class immunoglobulins needed for that infection. Then they make plasma cells that will release IgM if they do not have this helper T cell help. Right, and then one more fact is that the T cells and B cells can bind to the same antigen, but the epitopes that they use to bind to can be different. Real quick here, recirculation of helper T cells. Remember that the naive T cells come from the blood and they are bound to the antigen. If they do get activated, they are retained in the lymph node for a longer time. And then once they have increased, they proliferate and make a clone, they'll exit the lymph node and activated T cells will go to the periphery. If they continue to be naive T cells, they will move from one lymph node to another, which is called T cell recirculation. Couple of differences from the lymph node. There are other secondary lymphoid organs, such as Peyer's patches in the gut, that have HEVs from where the lymphocytes enter, but the antigen comes from the gut. And from the gut, they enter the lymphatic tissue using M cells. So M cells have no villi, no mucus coating. They bind to the antigens in the gut and transport that antigen from lumen to the tissue into the lymph node. And then some of the antigens also come opsonized with complement or antibodies. Another one is spleen. Spleen is a blood filter, which means it doesn't need HEVs. It's an open filter where the lymphocytes can enter freely into the spleen, but the antigens can come. So there are no incoming lymphatics because the spleen is a filter for blood. It's taking care of all the antigens that are in the blood. And there are no HEVs because the lymphocytes can enter freely in there. Here, there are B cell areas and T cell areas. The B cell area, it has the follicles, and the T cell area is called periarterioral lymphatic tissue. So it's called PALS. So remember, PALS is the T cell area and the lymphoid follicles are the B cell area in the spleen. What is the function of the secondary lymphoid organ? It has a strategic position where it can help in mobilization of different weapons to the site of infection and helps in interaction of these different components, lymphocytes, antigens, antigen presenting cells. A couple of more slides. We did talk about how the lymphocytes help in moving one cell from one place to another. How do these cells know where to go? The naive lymphocytes have unrestricted movement from one lymph node to another, and the way they end up in from one lymph node to another, because they express particular adhesion molecules such as L-selectins or some integrins like alpha 4 beta 7 that binds to its interacting protein or a surface molecule called MEDCAM1. So this is an important interaction to remember: alpha 4 beta 7 to MEDCAM1 and L-selectin to glycam1 that helps in lymph node homing. Depending on what it expresses, it can home to the gut or skin or various different lymph nodes, such as alpha E beta 7 binds to adresin helps in taking that lymphocyte to inflamed mucosal blood vessel. All right, that was the end of